Hi, hello. Um, Good morning or good afternoon, everyone. I'm Hironori Nakagami in Osaka University in Japan. Um, Today's Japan Society of Gene and Cell Therapies organized this web seminar. Our society is now, fortunately, is getting bigger and bigger. Now 600 to 700 members, but getting increase every year. So because a lot of the basics scientists and also clinicians is more interested in the field of gene therapies now. Okay, so today's our society is has two excellent speakers. The first, I will introduce the first speakers, Saki Kondo Matsushima, GK University, GK School of Medicine. So she graduated from the Kitazasu University and also got PhD in the University of Tokyo. Now she moved to the GK University School of Medicine. So that is a very famous private medical school in Japan and also very active in the field of gene therapies. So today's her title is AAV gene therapies uh, en- enabling the CNS delivery via BBB penetrating enzymes for lysosomal storage disorders.
Thank you for your kind introduction, Dr. Nakagami. Hello, everyone. I'm Saki Matsushima from the JK University School of Medicine. Today, I'd like to present our work on AAV-based gene therapy for the treatment of GM1 gangliosidosis. In particular, I will focus on our strategy to enable delivery of therapeutic enzymes into the central nervous system. Lysosomal storage disorders, LSDs, are inherited metabolic diseases caused by enzyme deficiencies. These deficiencies lead to substrate accumulation within lysosomes, resulting in progressive cellular dysfunction and tissue damage. Importantly, many of these disorders affect the central nervous system, leading to severe neurological symptoms. Because neurons are particularly sensitive to lysosomal dysfunction, even small levels of substrate accumulation can lead to irreversible damage. Therefore, early and effective intervention is essential. These disorders are particularly suitable for gene therapy due to the cross-correction mechanism. The enzyme produced in transduced cells can be secreted and taken up by neighboring cells via the mannose-6-phosphate receptor. This is a key advantage when designing gene therapy strategies for lysosomal storage disorders. This means that even partial transduction can result in widespread therapeutic effect. This is particularly important in the CNS, where achieving high transduction efficiency in all cells is challenging. GM1 gangliosidosis is caused by deficiency of beta-galactosidase, beta-gal. This enzyme is responsible for degrading GM1 ganglioside, which is particularly abundant in the brain. As a result, GM1 accumulates in neurons, leading to progressive neurodegeneration. In the most severe infantile form, patients die within the first few years of life. Currently, there is no effective treatment for CNS manifestations. AAV-mediated gene therapy is a promising strategy, and several AAV-based therapies have already been approved worldwide. Many of these therapies are administered intravenously, demonstrating the possibility of systemic delivery. However, efficient delivery to the CNS remains a major challenge. Intravenous administration is less invasive and effective for systemic symptoms. However, therapeutic enzymes do not efficiently cross the blood-brain barrier, BBB. This is one of the major limitations of current enzyme replacement therapy. Even when high levels of enzyme are present in circulation, only minimal amounts reach the brain. Therefore, CNS symptoms remain largely untreated. This gap between systemic efficacy and CNS efficacy represents a critical unmet medical need. Since GM1 ganglioside is abundant in the CNS, targeting the brain is essential. Therefore, we aim to overcome this limitation by developing a BBB penetrating enzyme strategy. As you can see, the BBB tightly regulates the entry of large molecules into the brain. Large proteins, including therapeutic enzymes, are generally excluded from passive diffusion. To overcome this limitation, we utilized transferrin receptor-mediated transcytosis. This is a physiological pathway normally used for iron transport. We fused an anti-transferrin receptor antibody to beta-gal, generating a BBB penetrating enzyme, which we call T-beta-gal. This allows the enzyme to bind to endothelial cells and be transported across the BBB into the brain parenchyma. The transferrin receptor is widely expressed throughout the body. This widespread expression is advantageous because it enhances uptake not only in the brain, but also in peripheral tissues. Therefore, this approach may improve both CNS and systemic efficacy simultaneously and may reduce the required viral dose. We designed AAV vectors expressing either P-beta-gal or beta-gal alone, and as a liver-specific promoter. The liver acts as a production site, continuously secreting the enzyme into circulation. This enables systemic distribution to peripheral organs and the brain. In the control AAV-treated group, the enzyme circulates in the bloodstream and is delivered to peripheral organs, whereas in the T-beta-gal treated group, it is delivered not only to peripheral organs, but also to the central nervous system by crossing the BBB. First, we evaluated enzyme activity. Both groups showed increased activity in peripheral tissues. And here, and here. However, only T-beta-gal showed significant activity in the brain. This clearly demonstrated successful BBB penetration. Next, we evaluated GM1 ganglioside accumulation in different brain regions. In untreated and beta-GAL-treated mice, substrate accumulation remained high. In contrast, T-beta-GAL treatment resulted in near complete normalization. Importantly, this effect was consistent across multiple brain regions, including the cerebellum and hippocampus. This suggests that the therapeutic enzyme is widely distributed throughout the brain. Such widespread correction is essential for treating neurodegenerative diseases. We also examined lysosomal pathology. Enlarged lysosome were observed in untreated mice. In contrast, the abnormality was markedly reduced in T-beta-GAL treated mice. This indicates correction at the cellular level. We next evaluated neuroinflammation. The GFAP positive reactive astrocyte and EVA1 positive activated microglia were significantly reduced. Neuroinflammatory is a key contributor to disease progression in lysosomal storage disorders. Therefore, suppression of inflammation is an important therapeutic outcome. In addition, inflammatory cytokines were also downregulated. This result indicated that our therapy suppresses neuroinflammation, suggesting that our treatment has both direct and indirect neuroprotective effect. We performed several behavioral experiments. First, we performed the rotor-rot test to assess motor coordination and balance. This test measures how long the mice can stay on a rotating rod. T-beta-GAL treated mice showed significantly improved performance compared to untreated and beta-GAL treated mice, indicating improved motor function. We also performed an open field test and measures the rearing number, which reflect exploratory behavior and general motor activity. T-beta-GAL treated mice showed a significant increase in rearing activity, suggesting improved neurological function. Finally, we performed gait analysis to evaluate walking patterns and coordination. I show you the typical example of each group, very fast and smooth. And it's untreated mice. Gait was impaired and irregular. The third one is beta-GAL treated mice. It looks similar to untreated mice. Whereas T-beta-GAL treated mice showed a more stable and coordinated walking pattern similar to wild-type mice. Like this. This indicates substantial improvement in locomotor function. Taken together, these results demonstrate significant functional recovery, not just molecular correction following T-beta-GAL treatment. T beta gal treatment dramatically extended survival. Untreated mice died around 300 days, whereas treated mice survived up to 600 days. This is one of the most important outcomes in this study. Survival extension reflects overall therapeutic impact, integrating all effect observed. This demonstrates a sustained and profound therapeutic effect and suggests that our approach has strong translational potential. Furthermore, earlier treatment resulted in even greater improvement. This highlights the importance of early intervention, especially in rapidly progressive diseases. We demonstrated an AAV-based gene therapy enabled CNS delivery via BBB penetrating enzymes through transferrin receptor-mediated transcytosis. This approach achieved robust CNS delivery and therapeutic efficacy at low dose following intravenous administration. This platform represents a promising strategy for the treatment of GM1 gangliosidosis and other lysosomal storage disorders, and has been selected for oral presentation at the 2026 annual meeting of the American Society of Gene and Cell Therapy. This therapeutic approach, which is the combination of AAV and T enzyme, is expected to be applied for the treatment of several hereditary neurological diseases with CNS involvement. Finally, I would like to thank all my collaborators for their support. Thank you for your attention.
Okay, so I have a couple of questions. The first question is looking at the distribution of transferrin receptors, they are also highly expressed in muscles and lungs. So were there any organs other than the central nervous system where this vector was highly expressed?
Yes, thank you for your question. We observed increased enzyme activity in peripheral organs, such as liver, spleen, and heart, reflecting the widespread expression of transferrin receptor. Importantly, we have also observed efficient delivery to skeletal muscle in other diseases models, suggesting broader applicability of this platform.
Okay, thank you very much. The second question is, this is a question regarding the dosage. Compared to the beta-gal alone group, it seems that the blood concentration and expression in the T-beta-gal group do not change much. Is it possible to reduce the dosage compared to conventional vectors?
Yes, we believe dose reduction is feasible, while the fusion slightly reduce enzyme activity. Peripheral expression remains comparable to beta-gal alone. Importantly, the T-beta-gal shows a clear advantage in the CNS, which is our primary target, so supporting the potential for lower dosing, we think.
It's great. So final question is about the effectiveness of the treatment. While early treatment is effective, when translating the stage estimated to be effective with this treatment to human, up to what age is it considered possible to treat in human?
Yes, early intervention is likely to be more effective, as seen in our mouse studies. However, we believe that treatment may still provide benefit even after disease onset, depending on the extent of neurodegeneration. So this will need to be carefully evaluated in clinical setting.
Okay, thank you very much. Again, congratulations on your success of your research. Thank you very much.
Thank you very much.
Okay, so next speaker is Kenichiro Kosai, professor in the Kagoshima University Graduate School of Medicine and Dental Sciences. So he graduated from the Kurume University School of Medicine and got PhD in the Kurume University School of Medicine. So he moved to the Baylor College Medicine, United States, and moved to Osaka and Kurume and Gifu. And finally, he is now professor in the Kagoshima University Graduate School of Medicine and Dental Sciences. So today's his talk is from basic to the clinical development of surviving M-CRA oncolytic adenovirus vector series.
Dr. Nakagami, thank you for introducing me and for chairing this session. I would also like to thank the organizing committee for this opportunity. Today, I will present the basic science behind our original Surv.m-CRA oncolytic adenovirus series and its clinical trials. This is my COI. In the early 1990s, we demonstrated that local immune gene therapy could induce systemic anti-tumor immunity. Subsequently, we began researching CRAs, conditionally replicating adenoviruses, which are another term for oncolytic adenoviruses. At that time, we hypothesized that a single factor in an oncolytic virus, including the CRA, would not be sufficient for precisely targeting cancer cells. So we developed a platform technology for the efficient construction of CRAs regulated and/or treating cancer with multiple factors, m-CRA. We use this technology to screen several cancer-specific promoters and identify surviving responsive m-CRAs, sub-m-CRA, as one of the best. We completed 14 human clinical trials of sub-m-CRA1 without transgene, and the phase 3 study is underway. Furthermore, 2 non-clinical studies of sub-CRA2 armed with immune genes are currently underway. The construction of diverse types of highly modified CRAs in large numbers at a time remains a time-consuming and labor-intensive process. So, to address this issue, we initially developed a platform technology to efficiently construct diverse m-CRA constructs. The genetic elements, including viral replication, therapeutic gene, adenoviral vector backbone, were introduced into 3 plasmids. This allows for unrestricted construction and fusion of individual elements, facilitating efficient generation of diverse types of m-CRAs concurrently. So E1A is a predominant determinant of adenoviral replication. Therefore, to generate the best m-CRA backbone, we first screened several cancer-specific promoters for regulating E1A expression. For example, we analyzed the TART and E2F cancer-specific promoters. These promoters were ideal because activity was detected in G0 face cancer cells, but nothing face in normal cells. However, the short-term HTART promoter is more active than the long-term promoter, which is consistent with the original biological paper. Therefore, it is important to carefully re-estimate backbone to identify the MCRA backbone in the actual adenoviral vector with the best performance. We found that survivin expression is high in most type of cancer, and interestingly, correlate with a poor prognosis in cancer patients. Both promoters were undetectable in normal cells. However, the best surviving promoter region was more active than the best TART promoter region. Consequently, Surv.m-CRA proved to be more cancer-specific and effective than TART-m-CRA. Deleting the RB binding site in Surv.m-CRA did not result in any significant phenotypic differences. In contrast, when mutated E1B was regulated by another cancer-specific promoter, cancer specificity increased significantly without decreasing anti-cancer effect. However, we ultimately decided against using the E1B promoter alteration for the first clinical development because from a commercialization standpoint, we would like to target all cancer types with the first product Surv.m-CRA1. In accordance with the clinical finding, survivin promoter is furthermore upregulated in cancer stem cells. As a result, Surv.m-CRA not only kill all populations efficiently, but also demonstrated increased therapeutic efficacy against cancer stem cells that are resistant to conventional chemoradiotherapy. These figures summarize the features of Surv.m-CRA1, which does not contain a therapeutic gene and was used in our clinical trials. In another project, we discovered that survivin promoter is highly active in undifferentiated and tumorigenic human pluripotent stem cell HPSCs. Notably, the Surv.m-CRA not only kill differentiated HPSC, leaving differentiated normal cells unharmed. These results demonstrate the high tumor specificity of the Surv.m-CRA. So, based on this result, we conducted a non-clinical study and phase 1-2 clinical trials. A phase 3 clinical trial for approval is underway. The GMP product was manufactured by an American CDMO. GLP non-clinical safety studies were conducted at American and English CROs. The regulatory consultation in Japan prior to the first in-human clinical trial took place between 2014 and 2015. So this is a clinical protocol for our first in-human phase 1 clinical trial. Patients with advanced malignant tumor in the bone and soft tissues received a single intratumor injection of Surv.m-CRA at 1 times 10 to the 10th, 11th, and 12th viral particles. Patients were eligible if they were over 9 years old and ineligible for standard therapy. The primary endpoint is adverse and side effects. We also assessed the local response rate as a secondary endpoint using the Choi criteria, which employs the enhanced CT as well as the popular RECIST criteria. The first patient with a recurrence of a sacral chordoma enrolled in this clinical trial. Under ultrasound guidance, she received a low dose of Surv.m-CRA. 28 weeks after viral injection, a nearly 50% reduction in tumor size was observed, and the anti-tumor effect persisted over for 2 years. The second patient, who had a recurrence of sacral chordoma in the vertebrae, also received a low dose of Surv.m-CRA via CT-guided intratumor injection. 40 weeks after treatment, CHY-based tumor necrosis exceeded 30% for more than, and the anti-tumor effect persisted for more than 3 years. Interestingly, beneficial bone remodeling, which occurs only with tumor death, was observed. Patient is currently alive 8 years after treatment. Similarly, we observed the response in 6 out of 9 patients. There were no serious adverse events directly related to the drug in any of the patients, including those in the high-dose group. Subsequently, we conducted a multi-center phase 2 clinical trials of Surv.m-CRA, focusing on the treatment of malignant bone tumors only, except for soft tissue sarcoma for future approval. We administered 5 intratumor injections of 1 time 10 to the 11th viral particle, with each injection 4 weeks apart. The primary and secondary endpoints are essentially the same as in the phase 1 study. This is a summary. Based on the Choi criteria, it is assumed that the response rate to other treatment for primary bone tumors ranges from the few percent to 20%. According to the Choi criteria, the Surv.m-CRA phase 1 clinical trial observed a response rate of 67%. The clinical study reports for the Surv.m-CRA phase 2 clinical trials in progress. After consulting with the regulatory authority, PMDA, we have moved on to phase 3 clinical trials in Japan. So, a phase 3 clinical trial is currently underway in Japan to obtain regulatory approval for the commercialization of a treatment for malignant bone tumors. The patient criteria and interventions are the same as in the phase 2 studies. The most important point is that based on regulatory consultation and the approval, the primary endpoint changes from RECIST to the Choi criteria. The Choi criteria demonstrated a clear therapeutic effect in phase 1 and phase 2 trials. I will now change the subject to discuss the basic research on the second generation Surv.m-CRA2, for which non-clinical trials are underway. So we generated various types of Surv.m-CRA2, each armed with different immune genes downstream of different promoters. Additionally, we established a syngeneic tumor model in hamsters because mice are not permissive for human adenovirus replication. Long ago, we demonstrated the optimal transgene expression levels are crucial for cytokine gene therapy. Only the optimal dose produces the minimal side effects and maximal therapeutic effects. Furthermore, the transgene is in oncolytic virus amplifies erratically as the virus multiplies in the cancer cells. Therefore, we speculated that careless oncolytic virus immunotherapy may induce excessive cytokine circulation and subsequent side effects. We constructed 4 Surv.m-CRA2s that either express no transgene or express a mouse GM-CSF transgene under the control of the E2F, RSB, or CA promoters. To examine systemic and parental cancer-specific immunity after viral injection, hamsters were inoculated with tumorigenic dose of 2 cancers, renal or parental pancreatic cancer, parental renal and other pancreatic cancer. All hamsters treated with Surv.m-CRA2 expressing mouse GM-CSF showed complete protection against parental cancer challenges. The immune reaction was tumor-specific to the parental cancer. So in contrast, hamsters treated with Surv.m-CRA's CA-PGM, which was the strongest promoter, exhibited systemic and lethal GM-CSF circulation and showed the overall survival. Autopsy reveals severe inflammatory reactions and injury to multiple organs. For our next project, we are trying to identify the optimal combination of immune stimulatory genes for the new Surv.m-CRA in the same experimental systems. This is just a summary. Based on our expertise and preliminary experiments, we identified 3 ideal genes, a 2 or 3 agents combination of Surv.m-CRA2 armed with either A, B, or C transgenes drastically increase the therapeutic effect against the primary tumor compared to the monotherapy. Similarly, a 2 or 3 agent combination A, B, and C transgenes significantly increase the parental tumor-specific and systemic immune response compared to the monotherapy. This was evaluated using a distance challenge test 2 weeks after a local intratumor injection. This figure demonstrates the proposed mechanism of action of the three agent Surv.m-CRA2 therapy. Transgene A induced immune cell infiltration. B stimulate antigen presentation. C also activate T cells, especially cytotoxic T lymphocytes. So finally, Surv.m-CRA2 IC immune gene complex dramatically induce systemic anti-tumor immunity. So this is a conclusion. Following the positive result of phase 1 and phase 2 trials, we are currently conducting the phase 3 clinical trial of Surv.m-CRA with the transgene to treat malignant bone tumors in preparation for approval in Japan. We are conducting non-clinical study on 2 products of Surv.m-CRA2 that are armed with immune stimulatory genes. The optimal promoter is essential for achieving maximal safety and maximal selective effect with Surv.m-CRA2G, GM-CFG, and the several promoters. Surv.m-CRA2-IC3 combination, which is armed with 3 transgenes, significantly enhanced systemic anti-tumor immunity, inhibited primary tumor growth, and prevented distant metastatic formation. Kagoshima University, National Cancer Center, and Kurume University participated in the phase 2 multicenter clinical trial, and the Tokyo Institute of Science is supporting statistical analysis. The university-affiliated startup is also providing support for the current phase 3 clinical trials. I would like to acknowledge all the collaborators involved in this project. I would like to acknowledge the contributor of the members in my laboratory and the financial support from public grants provided by the Japanese government. Thank you very much for your attention.
Okay, so you present a lot of data, but I summarize into 2 simple questions. The first question is regarding the ongoing clinical trials. So why was primary bone sarcoma bone tumor selected for the first clinical trials?
Oh, thank you for the good question. This is a very important point. So the regulatory authority, the PMDA, suggested that for the first human trial, we should administer the drug either externally or to internal organs via endoscopy, either one. So therefore, we selected malignant tumors in bones or soft tissues for the first trial, and fortunately observed the positive results, as I demonstrated in my presentation. So for the phase 2 trials, as you know, we have to focus on one tumor type to aim for future approval, commercialization approval. So the phase 2 began as an investigator-initiated clinical trial that initially supported by the governmental fund. So malignant bone tumors are an example of both unmet medical needs and an orphan disease. So this is advantageous because a small number of patients need to be enrolled. In this regard, we have proceeded with the phase 2 and 3 clinical trials to treat primary bone tumors and seek approval. Currently, we are conducting the phase 2 trial for metastatic bone tumor also, and have completed the phase 1-2 clinical trials for pancreatic cancer. So if I have a chance, yeah, we'd like to present other trials. as well. Thank you very much for the question.
Okay, so the next question is, I guess that there are several hurdles from basic research to clinical trials, but what has been the biggest obstacle so far?
Yes, thank you for the question. Yeah, the biggest obstacle is the beginning of the non-clinical development. Also, I am trained as a pediatrician or pathologist, and I have devoted most of my career to the basic science of gene therapy or biological molecular therapy, molecular biology, biology. But I began conducting the non-clinical study almost in 2007. At that time, I had no experience in GMP manufacturing, including CMC, nor in GLP non-clinical safety studies of such specialized viral drugs. And also my small university had no resources, so I had to perform all of the non-clinical development by myself at the initial step. So it was biggest obstacle. Yes, thank you for the question.
Okay, so I will close this session. Today's two excellent speakers had very nice presentations. Actually, our annual meeting this year will be scheduled in the July in Osaka. So if you have a chance, please join us. Okay, thank you very much today.